Good morning, everyone, and thank you for having me here at this wonderful location, sharing some of our experiences with you at the ALAD Congress 2019. I'm quite delighted to be here, and I look forward to learning a lot and sharing a lot with wonderful colleagues and experts in different fields related to diabetes and cardiovascular risk. The topic that I have at hand is the role of ApoC-III as a cardiovascular risk factor. And I wanted to include in this slide the physical structure of ApoC-III, because I find it really interesting how such a small and relatively simple protein may have such profound effects on cardiovascular risk and how it may be such a great pharmacological target. To start, I want to point out that besides their constituent apolipoproteins, which are essentially ApoB and ApoA, different types of lipoproteins may or may not have on their surface this sort of small interchangeable apolipoproteins, and they are ApoC-III and ApoE. So I'm going to display 4 different types of lipoproteins. Let's assume this is an ApoB lipoprotein, so it could be VLDL or LDL. And then we'll have lipoproteins without ApoC-III, lipoproteins with ApoC-III, which we will display here in yellow, and lipoproteins with both ApoC-III and ApoE, which we will display in blue. So over the course of the presentation, we will be discussing the specifics of the function and characteristics of each type of lipoprotein and how they can be targeted for cardiovascular prevention. So let's start by looking at the in vitro evidence that ApoC-III acts as a competitor to ApoE and that it impairs clearance of VLDL and LDL. So what you will see here are the results of an in vitro receptor binding experiment in which different progressing concentrations of any given lipoprotein were incubated with fibroblasts in vitro. What we can see here is that the top line here, the white squares, corresponds to lipoproteins with only ApoB. The black circles are lipoproteins with ApoB and ApoE, and then the black triangles are the lipoproteins with ApoB and ApoC-III. And you can see that at any given concentration of the given lipoprotein, the binding is lowest for lipoproteins that contain ApoC-III. So since 1995 this evidence was published, so we know very well that ApoC-III impairs the clearance of VLDL and LDL. Not only that, but ApoC-III has a metabolic impact. It accelerates the production of dense LDL. And we all know that small dense LDL are particularly atherogenic, and they correlate very strongly with cardiovascular risk over the long term. So what we're seeing here, and we published this in 2009, is that lipoproteins containing ApoC-III and not any of the other small apolipoproteins tend to be converted to light LDL much faster than other types of lipoproteins. And then that's particularly true for light LDL. So in the step that goes from light to small LDL, the step that generates small dense LDL, the speed of production is increased by ApoC-III. Not only that, but ApoC-III in and of itself is able to induce atherogenic changes on the arterial wall, particularly on the endothelium. It promotes the recruitment of immune cells and their migration and differentiation to macrophages. So what we are going to see here, this is evidence from Akio Kawakami, it was published in Circulation 2006, is an experiment in which monocytes were evaluated for their ability to adhere in vitro to endothelial cells in the presence or absence of different types of lipoproteins. So we have the control situation here with no lipoproteins. This is with VLDL that do not contain ApoC-III. You see it is pretty much the same as the control situation, but if you add VLDL that contain ApoC-III, that adhesion almost doubles, signing up that ApoC-III and lipoproteins with this kind of apolipoprotein greatly increase the potential of macrophages to adhere to endothelial cells. And the same was true, even though to a lesser extent, for LDL containing ApoC-III. This is what we see here. Then the protein itself, purified, not as a part of a macromolecular lipoprotein, but pure ApoC-III was also able to induce an increase in monocyte adhesion. So we know that ApoC-III increases the production of small dense LDL. It increases the adherence of monocytes to endothelial cells. And not only that, we can see here that the response, the protective anti-adhesive effect of HDL is lost when they have ApoC-III. So this is the same experiment, but now we're incubating the monocytes not with VLDL or LDL, but with HDL. And you can see here, this is the control situation. When you add HDL that contains ApoC-III, there's pretty much no change, a very small, very mild change. However, if you add ApoC-III that does not contain ApoC-III, the adhesion goes down by about 40%. So in a way, the ability of HDL to inhibit the adhesion of monocytes to endothelial cells is dependent on the presence or absence of ApoC-III, or is highly correlated with the presence of ApoC-III. In the same set of experiments, the endothelial cells, and looking for mechanisms that could explain the monocyte findings, the endothelial cells in the presence of ApoC-III or ApoC-III containing lipoproteins express more adhesion molecules, particularly ICAM1 and VCAM1, and you can see here with increasing concentrations of purified ApoC-III, the expression, the surface expression of these two adhesion molecules goes up. And I want to highlight that the usual concentrations of ApoC-III in human plasma are in the order of 80 to 100 micrograms per ml, so this is physiologically relevant concentrations. However, the big boom about ApoC-III came when in 2008 a paper was published in Science, and it was sort of a natural experiment in which humans, and these individuals were part of the Lancaster-Amish community, individuals with a genetic ApoC-III deficiency, it was a nonsense mutation in the ApoC-III gene, they have reduced atherosclerosis and actually a reduced lifetime risk for cardiovascular disease. So what we have here is the data for individuals with the wild-type CC genotype, the mutant or the minor allele CT, and then we have the odds ratio for each outcome. So if you look at any coronary artery calcium, the odds ratio was 0.35 for people without ApoC-III. So there was a 65 reduction in the probability of having any coronary calcium. And when we look at the probability of having a CAC score greater than 100, the odds ratio was 0.40. So that's 60% less risk of a calcified large lesion in your coronary arteries. So this really draws a lot of attention to ApoC-III as a pharmacological target, and not only as a pharmacological target, but also as a major player in the regulation of lipoprotein metabolism and cardiovascular risk. So if you look at this from an sort of an biochemical / epidemiological point of view, and you try to look at whether or not the presence of ApoC-III signals the atherogenicity of lipoproteins, this is what you will find. So this was published by us back in 2011 in Circulation, and what we did was we measured LDL, but we separated LDL with or without ApoC-III, and we divided. This was a nested case control study, part of the HPFS and NHS cohorts, and we compared the cardiovascular risk across quintiles of LDL with or without ApoC-III, and what we found was actually pretty remarkable. Across quintiles of LDL without ApoC-III, there is no real trend to an increased risk, cardiovascular risk. But if you look at the quintiles of LDL with ApoC-III, there is a monotonic, consistent, robust increase in risk with every additional quintile. So this data could be interpreted as saying that a large proportion of the cardiovascular risks that we commonly attribute to LDL may in fact be due to a subtype of LDL that contains ApoC-III and not to all or any type of LDL. So when you adjust it for other important lipid and non-lipid cardiovascular risk factors, what we found is even after adjusting the association persisted. This is adjusting for LDL cholesterol, which is really important. Adjusting for plasma ApoB, it remained significant. And this is really interesting. It happened only for LDL with ApoC-III, that it was consistent, the association, even after adjustment for LDL cholesterol or plasma ApoB. In the case of LDL without ApoC-III, when you adjust by LDL cholesterol or by plasma ApoB, the association either is negative or actually turns to the protective direction. This is because the only way to have more LDL without ApoC-III having the same plasma ApoB is by having less of the bad type of LDL with ApoC-III. Then one step further, we analyzed, this was done by Mack and Jensen, it was published in YAHA in 2013, the association between HDL with ApoC-III or HDL without ApoC-III. So it was kind of a parallel of what we did for LDL, but now with a protective HDL. What we found was HDL with ApoC-III completely loses its protective ability or protective prediction, whereas HDL without ApoC-III was, as we would expect for HDL, monotonically associated with protection or with negatively associated with cardiovascular risk. So how could we explain this? When we look at the binding of VLDL to cellular receptors and we separate VLDL with ApoE and with ApoC-III, this is what you see. When you add ApoE, there's pretty much no change, but when you add ApoC-III, the more ApoC-III molecules you have per particle, the less binding to receptors you have. Now, could this be reversed or somehow opposed by the other type of small interchangeable lipoprotein, that is by ApoE? Since they are mechanistically antagonic, could they also be antagonic at the epidemiological level? That's what we asked in this study. And what we found pretty much confirmed our hypothesis. So across quintiles of ApoE, and this is ApoE in molecules or in particles that contain ApoC-III, there is less and less cardiovascular risk. So in a way, there is this constant competition, this yin-yang between ApoC-III and ApoE in balancing the risk imparted by ApoB containing lipoproteins. So we wanted to look deeper into the mechanisms responsible for these opposite actions of ApoE and ApoC-III in lipoprotein metabolism in general, but particularly in HDL metabolism. So what we looked at was at how the interaction of ApoE and ApoC-III influenced the in vivo metabolism of HDL in a kinetic study. And this is the general design of the study. We took 18 overweight or obese adults. We fed them for 4 weeks a controlled diet. Then after that, we interned them and provided them with a D3 leucine bolus. And we collected plasma serially for 46 hours. From that plasma, we separated it into 4 different types of HDL, so containing no ApoE and no ApoC-III, either one or both. And then we separated those HDL subtypes by size, and we did that here but not denaturing page. And then we transferred this to a membrane, we extracted the proteins, we hydrolyzed the ApoA, and then we measured the leucine, the D3 leucine enrichment in ApoA of each subtype. Then we modeled this mathematically using SAM, and we came to a biological model of how HDL works, and particularly how the different subtypes in terms of size and apolipoprotein composition interact with each other to get to an overall map of HDL physiology. So this is probably the most remarkable finding from this study, and this is when we observed the fractional catabolic rates of each type of HDL. So the fractional catabolic rate is the speed at which this particular type of lipoprotein is being regenerated and catabolized. So we assume steady state, so we assume the amount of production and the amount of catabolism are equal, and the fractional catabolic rate is just telling us how fast this is happening. We have the 4 sizes here at the bottom, And you can see that for every size, the highest bar is the one with the blue dots, which corresponds, as you can see here, to HDL non-containing ApoC-III but containing ApoE. So this particular type of HDL has a very, very fast metabolism. It is converted very fast. It can yield part of its cholesterol ester content faster than other types. They are produced faster and they are retaken faster than every other type of HDL. And then in the same publication, we looked at this from an epidemiological point of view, and this is the result from a nested case control study in a Danish, it's actually a case cohort study in a Danish large cohort, measuring again the subtypes of HDL and associating the concentration of each type with the risk of coronary heart disease. And again, the protective association was only observed for ApoE in HDL not containing ApoC-III. So this is a BC slide, and I'm sure I also want to encourage you to go and read the paper, but what we have here is a sort of a map of how each different size and apolipoprotein type of HDL is produced, is metabolized, and eventually retaken up. And how is the situation when you have a higher risk of CHD, and how is it when you have a lower risk of CHD? So trying to expand on these findings, we then looked at how dietary factors, particularly dietary unsaturated fat, may affect this whole map. Will it increase the metabolism of certain type of HDL? Will it decrease the metabolism of some other type? Will it increase the concentration or the distribution of some type of HDL? And this was published actually this year in JCI Insights. What we found is that, as we expected, a high polyunsaturated fat diet induced a greater concentration and a faster catabolism of HDL containing ApoE, corresponding here to the green dots. You see in every size fraction, the green dots are above the blue dots, pointing that ApoE containing HDL is metabolized faster, and that was more prominent or more pronounced in the high-fat diet. So now we're trying to look at the influence of ApoE and ApoC-III on the enzymatic activities of important enzymes for HDL function and reverse cholesterol transport. So two of those enzymes are LCAT, lecithin cholesterol acetyltransferase, and CETP, cholesterol acyl transfer protein. They are in charge of sterifying cholesterol and eventually exchanging cholesterol triglycerides with other lipoproteins. And what we did here, this has been submitted, it's under review at the moment, is in each type of HDL defined by the presence or absence of E and C3, what happened to the triglyceride, cholesterol, phosphatidylcholine, cholesterol ester concentration, and to the enzymatic concentration and activity of LCAT and CETP. So, in summary, what we found is this particular type of HDL that has the bad guy, that's ApoC-III, but does not have the good guy, that's ApoE, has a much larger triglyceride concentration. So it is, in a way, structurally dysfunctional. And when we looked at the enzymatic concentrations and activities, this type of E-containing but not C-containing HDL, so this one, even though it has a relatively low concentration of LCAT, it has an activity that is comparable to the dominating subtype, that is the type that has not E and does not have C3. So in a way, this could be interpreted as saying that HDL with ApoE but without ApoC-III has a hyperactive type of LCAT, some kind of associated factor, or maybe the apolipoproteins themselves influence the activity of LCAT so that with lower concentrations they achieve the same enzymatic activity as other fractions, which is in line with what we found in the kinetic studies. So lastly, this looks like a wonderful therapeutic target. And in fact, there is a drug, and antisense oligonucleotide, currently submitted for approval to several regulatory agencies, and its brand name is volanesorsen, or it's the generic name is volanesorsen. The brand name is Waylivra, and it is directed against the ApoC-III messenger RNA. So, just a quick overview. Antisense oligonucleotides penetrate the membrane, then they penetrate the nuclear membrane. They may act at the cytoplasm or at the nucleus. They bind by base pairing to the mRNA that is being targeted, and they induce their degradation by the RNase H mechanism. So there's been a lot of development of these kind of molecules in lipid metabolism, but particularly against ApoC-III, and volanesorsen was recently tested successfully, actually, in a monogenic condition called familial chylomicronemia. So in this case, the problem is usually a mutation in lipoprotein lipase. There is actually a gene therapy to try to replace lipoprotein lipase, but another option is to try to suppress ApoC-III and increase the uptake of triglyceride-containing lipoproteins, and or increase the activity of whatever LPL is left after the mutation. And this was recently published in the New England Journal. And what you see is this agent that targets ApoC-III was able to induce, and I have to highlight and I have to emphasize, this was only in patients with chylomicronemia, which is a fairly rare disease. It induced about a 70 to 80% reduction in plasma triglycerides that was maintained over a 52-week period. So it was really a major reduction. When you look at it numerically, the average reduction was 1,712 milligrams per deciliter of plasma triglycerides, which is really impressive. Now we have to take into account that these patients may have triglycerides in the order of 3, 4, 5, 10,000, but still, this is a major improvement. And this is just a press release from the manufacturer announcing that Waylivra, that is volanesorsen, was approved for use in the European Union. So I'm sure many other regulatory agencies may follow suit in the coming years. So this is what I wanted to tell you about ApoC-III, how it influences lipoprotein metabolism, how it influences the pathogenesis of atherosclerosis, how it constitutes a wonderful therapeutic target, and how it in fact has been targeted by a biotechnological drug successfully. So I hope to get you interested. I hope you will go and read the papers. And again, thank you very much for your attention. Have a great day.